Hello, my name is Jamel Popola. My group presentation is on genetic interference by double-stranded RNA in C. elegans. One of the main authors of the article is Andrew Fire. Andrew Fire is an American biologist who earned his undergraduate degree at the University of California in mathematics at the age of 19. He earned his PhD at Massachusetts Institute of Technology in biology. His research, which is on gene regulation in C. elegans using double-stranded RNA interference, was done at Carnegie Institute of Washington. He's currently a professor at Stanford University in pathology and genetics. Craig Mello, who is in charge of the lab, is an American biologist as well, who's a professor at the University of Massachusetts in molecular medicine. He earned his undergraduate degree at Brown University in biochemistry and molecular biology. He completed his graduate studies at the University of Colorado in molecular, cellular, and developmental biology. He earned his PhD at Harvard and completed his postdoc at Fred Hutchinson Cancer Research Center. C. elegans are a member of the Nematoda family, and they're a form of roundworms which share many essential biological characteristics with human, despite how primitive they are. Their complex embryonic developmental system has been studied throughout the years and has been used in multiple research. Some of the C. elegans genomes has functional counterparts to humans' genome. For example, the Myidae family, which was talked about in the article. C. elegans mutants can be studied to better understand human disease. For example, mutants with neurological disorders, heart diseases, as well as kidney disease. Sense versus antisense. The sense is the codon strand, and the antisense is the non-codon strand. The sense strand runs in the 5' to 3' direction and is complementary to the antisense strand, which runs in the 3' to 5' direction in this figure. The antisense strand is considered a template strand, and it contains all the information necessary for the mRNA synthesis. The sense strand has a similar base sequence to that of tRNA. So nuclear localizational signals allows protein to go from the cytoplasm to the nucleus. So this can only happen after translation of a protein and after folding of the protein. So proteins that are going to the nucleus have a nuclear localizational signal, which is a certain amino acid sequence, and it's going to bind to receptors called importans, which are found in the nucleus. These importans are going to attach to cytoplasmic fibrils because they have affinity for FG repeats on the cytoplasmic fibrils. They're going to bind on and off. The cytoplasmic fibrils and move down in a zigzag-like motion, which the directionality is given to them by RAN-GTP. Once it gets into the nucleus, the importance is going to release the protein with the signal peptide to get into the nucleus, and it's going to bind to another protein, and the process is going to start all over again. With mitochondrial localizational signal, the protein can only enter the mitochondria after translation, but before folding. So in order to prevent misfolding of the protein, the hydrophobic regions are going to be covered by HSP70. HSP70 only provides an environment for the protein to fold correctly, but in this case, the protein isn't going to fold until it's completely into the mitochondria. So what happens is HSP70 is going to bind and release the protein while it travels through the mitochondria. First, it's going to go through the Tom complex, and once it gets to the Tom complex, cytoplasmic HSP70 is going to release it, and it's going to get fully into the Tom's complex, and it's going to be into the outer matrix once it's past the outer mitochondrial membrane. Because ATP is needed for this process to happen, the binding and releasing of HSP70 from the cytoplasm is going to provide energy, as well as the electrochemical gradient provides energy. Once in the mitochondrial HSP70 is going to continue helping the correct folding of the protein by hiding hydrophobic regions. In order to get into the inner matrix, the Tom and Tim complex have to align, and so it's going to travel past the outer matrix to the inner matrix, where it's going to be folded correctly. Once the protein is fully folded correctly with the help of chaperones, the signal peptide will be cleaved by signal peptidase, and you'll have the mature mitochondrial protein. Analysis of phenotypes. By observing physical characteristics of an organism gene, function can be determined. You can do this by using gene knockout, mutation, overexpression, or these are possible ways you can analyze phenotypes. By collecting various mutant strains, it will be possible to eventually shed light to gene function. In the article, the authors noticed that decrease in UNC22 activity led to increasingly severe twitching, while loss of function resulted in impaired mobility and structural defects. The use of GFP reporter genes. Reporter genes aid in understanding of signal pathways and gene expression. GFP was isolated from Aquaria victoria. GFP emits a green fluorescent light when exposed to UV light. GFP can be inserted into the gene of interest, and once that gene is transcribed and translated into protein, the GFP will be within that amino acid sequence. So whichever cell that gene ends up in, or the location the gene ends up in, it can be tracked under a microscope and then examined. This can help determine where the gene ends up after translation, or the location the gene could possibly determine function of that gene as well. LAGZ, the use of LAGZ as a reporter gene. The LAGZ promoter produces beta-galactosidase, and in the presence of XGAL, beta-galactosidase will cleave XGAL. This will create a blue precipitate. If you insert a DNA of interest into the multiple cloning site of LAGZ, this will cause LAGZ to be non-functional. If LAGZ is not functional, beta-galactosidase would not be produced. When beta-galactosidase is not produced, XGAL would not be cleaved. If XGAL is not cleaved, then no blue precipitate is formed. This lets us know that the gene of interest is functional and is being produced. Figure 1. Each gene in Figure 1 represents basically the starting point of the experiment. So UNC22 encodes a non-essential myosin filament. FEM1 is responsible for a protein required in sperm production. HLH1 encodes a protein required for proper body shape and motility, while UNC54 is responsible for full muscle contraction. The segments above represent genomic RNA interference based off genomic DNA, while the segments below represents RNA interference based off cDNA. So these are the area that is being regulated, where the A, C, and B are located. These are what's being regulated in each of these genes. And in the bottom, the tags are added to Myo3 so they can track where it ends up in the nucleus or the mitochondria. And they want to do this so they can increase specificity of RNA interference and allow for more gene regulation. My only critique was that the authors mentioned somewhere along the line that there was not all the data shown in their article. They're missing data about co-injection of double-stranded RNA in the UNC22 gene. So I was just wondering, why wasn't that there? Why wasn't it included in the article? And these are the citations, and that's the end of my presentation.